Another important mechanism for control of enzyme activity is by reversible phosphorylation. Let's first consider the general principles of this mechanism and then look at an important example. By phosphorylation, we mean the covalent attachment of a phosphoryl group to an amino acid side chain. The donor of the group is typically the nucleotide ATP, and the bond forms by attack on the phosphorus atom by the nucleophile within the side chain. Most commonly, this occurs with the oxygen atom in the hydroxyl groups of serine, threonine, or tyrosine. Phosphorylation is a very common mechanism and typically results in a significant change in enzyme activity. Phosphorylation of one enzyme may result in inhibition of activity, whereas the very same event in another enzyme may result in activation. Later, we'll look at phosphorylation in an example of membrane transport. Let's consider the example of glycogen phosphorylase. As you will learn later, this is a key allosteric enzyme that releases glucose molecules from glycogen stores. You can think of this enzyme as allowing us to open our cabinets and take out the food we need when current resources are limited. As is true for all allosteric enzymes, it has two conformations and can be influenced by various effectors to favor one or the other. It is also subject to covalent control through phosphorylation. Kinase enzymes attach phosphoryl groups, and phosphatase enzymes remove them. Control is therefore a balance between these two activities. Phosphorylation of glycogen phosphorylase converts it to the more active A form, as shown on the top right of the figure. Removal of this group converts it back to the less active B form on the top left. Notice from this figure that the two forms of glycogen phosphorylase, the A form and the B form, may each exist in either the R or T conformations. This means that each is subject to allosteric effectors, though they are different in each case. We'll look at this example in more detail later. For now, suffice it to say that the net effect of covalent modification and allosteric effectors in this case is that the active A form predominates when energy is needed from glycogen breakdown.